Hello everyone. Lecture 4 is going to be a tour of the cell where you're going to be able to describe the components of a living cell, the smallest unit of life. Recognize microscopic techniques, name cellular organelles that are going to be depicted in microscopic images taken by those techniques. And hopefully by the end you'll be able to not only identify the technique, but identify how the use of different techniques will help you to better see and understand the components of the cell. We'll also list and describe the macromolecular components of important cellular structures. This is a connection to lecture 3. We'll describe the function of cellular organelles and the structures and how the macromolecular composition of those allows for their given function. And finally, we'll explain how cellular organelles act cooperatively to achieve cellular functions. This really requires you to not only work with individual components, but start to connect those components together. And this fifth ability for you is going to be something that develops over time. So first, I'd like you to start by pausing the video and doing the 6 review questions on Socrative that you see here, to double check that you have a good understanding of the macromolecules that we're going to be using to assemble the components of the cell. All right, so what are the functional groups that we discovered, which is basic? That would be an amino group. And what monomer type contains this group? An amino acid. Now, amino acids can be linked together through dehydration synthesis to yield polypeptide chains. By the way, here's another question you could ask yourself. What's the enzyme that synthesizes that polypeptide chain? Good, that would be a ribosome, and that ribosome is an enzyme, so it's going to be made out of proteins. Now, what type of bond allows for secondary protein structure? Those are hydrogen bonds between non-neighboring amino acids. So the peptide bond that links amino acids one to the next, that actually forms the chain, that's going to be primary structure. That's what's assembled by the ribosomes. The quaternary level of structure is when we have interactions between multiple polypeptides shown to actually yield a functional protein. Now, this is your final question, and if you didn't have an opportunity to answer it, go ahead and do so now. What type of amino acid is shown? Now, in order to answer this question, you first have to identify the core structure of an amino acid. An amino acid has an amino group, one here, and note that there's a second one here. So that means that we can't just use that group in order to tell us what type of amino acid we have. We have to cross off the entire core. The second part of an amino acid is an acidic group. So which group do we know is acidic? That would be a carboxyl group. A carboxyl group is composed of both a carbonyl group and a hydroxyl group. Well, we have 2 carbonyl groups, one here, and we have one here. In order for it to be a carboxyl group, that's the acid of an amino acid, we have to have both a carbonyl and a hydroxyl together. Well, the only one that has a hydroxyl group right next to it, meaning that we have an actual carboxyl group, is going to be this one up here. So that means that the core structure of our amino acid is going to be this group right here. So in order to identify the type of amino acid that we have, we need to take a look at the rest of the molecule. Well, we have some hydrophobic bonds here, some non-polar covalent bonds. We do have a hydrophilic bond, some polar bonds here, but the fact that we have an amino group sticking out here at the end means that this is really going to give us a truly basic property. Now, in order for us to really start to understand the components of a cell, we first need to be able to view those components. Cells are too small to be able to see with the naked eye. So in order for us to be able to see them, we need to make use of microscopy. And the components of a cell, even though the cells themselves can be seen by light microscopy, which you've probably used in biology classes before, those individual components inside of the cell need to be magnified even further. So we're going to make use of electron microscopy to see in greater detail. Light microscopy uses the properties of light, which pass through various lenses, and bend the light in such a way so that when it reaches your eye, it's magnified. Light microscopy can allow us to view entire cells, but it can also allow us to view some of the components of that cell, such as a nucleus. Unfortunately, though, the cell itself and the individual organelles within that cell are still too small to be seen by light microscopy. If you guys are interacting in lab and using microscopes in the lab of this section, you're using light microscopy, but you're only going to be able to view very, very large things, entire cells, as well as possibly nuclei. So here's an example of an entire cell, which you can see under light microscope. This is a cheek cell that's been stained, and this structure in the middle right here is the nucleus. It's the largest organelle in the cell, and yet it's still something we can only see very, very small. This is known as bright field light microscopy. And you can tell it's a bright field microscopy because the background is actually going to be bright. So this is a field of view that's bright, hence it's a bright field light microscopy. You'll notice that bright field light microscopy has a lot of important things and that it can actually share with us. Here's an example of light microscopy, specifically bright field light microscopy, and one of its greatest advantages. And that is that we can use light microscopy to view live cells. This is known as cytoplasmic streaming. That's what you're seeing here. That's the cytoplasm inside of the cell, which is moving around, and what you're seeing, these green structures here, these are going to be chloroplasts, which are suspended in the cytoplasm that are moving around. Now, there are other kinds and types of light microscopy. Only one of them do I want you to know in addition to bright field light microscopy, and that's fluorescence light microscopy. Fluorescence light microscopy is distinctly different. The first thing that you'll notice is that the background of all of these images is black. The other thing you'll notice is that the structures of the cell are glowing, they're fluorescent. And that's because we are labeling using molecules that specifically bind to cellular components, to particular molecules inside of the cell. And that allows us to color only certain components inside of the cell. Fluorescent light microscopy, just like bright field light microscopy, is still only going to allow us to observe very large components, entire cells, such as the cytoskeletal network or the entire nuclei. But you're going to be able to distinguish some individual components because of the labeling. Electron microscopy, on the other hand, is going to give us additional information that's very important, simply because we're going to be able to see components much larger. Electron microscopy is going to be things that allow you to see individual organelles. Now, electron microscopy does not use wavelengths of light. Since light is what allows us to view color, that's why bright field light microscopy and fluorescence light microscopy images can be seen in color. Electron microscopy images are sometimes colored on a computer, such as the ones you see here, but anytime that I ask you to identify an electron microscopy images, it will be uncolored. So the very first thing you can do, the easiest way to help you identify different types of microscopy images, is to simply start by determining whether you're seeing light or electron microscopy. If you're seeing very, very high levels of detail, very largely magnified images, you already know you're dealing with electron microscopy. And if the image is in black and white, that's a good supporting fact for you to make sure that that's what you see. If you've already identified it as electron microscopy, the next thing you need to do is decide whether it's scanning electron microscopy or transmission. Scanning electron microscopy scans along the surface of your specimen. And so it allows you to see three-dimensional images because of the fact that we're actually scanning along the surface. So what you're seeing here is an example of cilia, which are protrusions on the surface of cells. Transmission electron microscopy allows the electrons to pass through the specimen. We actually cut the specimen into very thin slices. And so this is an electron microscopy image of those same cilia. But the difference is that scanning reveals the surface texture and transmission electron microscopy reveals the internal structures. So now what we're going to do is give you an opportunity to test yourself. All of these are images of neurons, but they've been taken using different types of microscopy. So now I want you to log into Socrative, pause it here if you need more time, and start by identifying which of these images was taken using fluorescent light microscopy. Hopefully you identified the fact that fluorescence light microscopy is going to be the type of image that reveals to you only large components of the cell, and that this fluorescence light microscopy has a black background. So the answer here would be D. This is an example of fluorescent light microscopy, where we're seeing the entire cell, and the details are revealed only by these little fluorescent molecules. What about if I asked you to identify a scanning electron micrograph? While electron microscopy allows you to view more details inside of the cell, so we know it's not D. B is only allowing us to see giant components of the cell. There's no individual details inside. And A is colored, so we know that that's not going to be electron microscopy. That has to be light microscopy. So that leaves us with either C or E. Which one of these reveals surface details? Yeah, C does. And so because we have this three-dimensional shape and all the levels of detail on the surface, we know that C would be an example of a scanning electron micrograph. Now, all cells are made of the same four basic components. And these are going to be a theme that we come back to over and over again. So I do expect you to understand and know what these four basic components are. On the outside of every cell is a plasma membrane composed of a bilayer of phospholipids. We'll talk some today about this plasma membrane, but we're going to come back to that because that's the theme of our next lecture. Inside, bounded by that plasma membrane, is what's known as a cytoplasm. That cytoplasm is filled with a gelatinous, semi-fluid substance known as cytosol. The word cytoplasm is telling us that this is the shape, the filling of the cell. That's what cyto actually means. Suspended within that cytoplasm will be ribosomes. That's the enzyme that synthesizes proteins. And somewhere inside of that cell, every single living thing has a genome that's composed of DNA. One important difference, however, is that when we're looking at eukaryotic cells, in addition to having additional complexity, they're going to have a DNA genome which is actually found inside of a structure known as a nucleus. So eukaryotic cells will have an additional membrane which actually encloses this DNA genome inside of it. Now, their genome is also going to be linear, which is different from the fact that prokaryotic cells, which includes bacteria and archaea, have a circular genome. Eukaryotes can have one or more segments of their DNA, and that DNA is going to be associated with proteins called histones. There will be other membrane-bound organelles. So the nucleus is one, meaning that there's a membrane surrounding and forming the structure of that nucleus. but we're going to talk about some other nuclei or some other membrane-bound organelles as well. And that cytoplasm is still going to be filling the region between the plasma membrane and the nucleus. So why do we care so much about the fact that eukaryotes have all these extra membranes? And quite frankly, why evolve this extra level of complexity? Well, when you subdivide a cell by using additional membranes, what you do is you actually increase the surface area of those membranes. And one of the things that we're going to be learning over the next 2 weeks as we really start to dive into the functions that cells can have is that membranes are not just walls. Membranes can also do jobs. And so eukaryotic cells are able to do so many different things and have so many different functions, in part because all of those membranes which subdivide the cell don't just create different, unique, isolated spaces, but they give functions to those spaces. Here's a perfect example. Meet cilia. Cilia not only are going to expand beyond the structure of this cell, but all of this surface area that encases and covers these cilia, those are going to be the actual surface area that's used to absorb nutrients from the food that you eat in your small intestine. Now the plasma membrane, so this is the membrane on the surface of the cell, is going to be a phospholipid bilayer. Every single cell has a plasma membrane. Eukaryotic cells just have other additional membranes. That plasma membrane acts as a selective barrier. And embedded inside of that membrane, there are going to be proteins and carbohydrates that give additional functions. And when we place these together, we get some additional properties to that membrane. So now what we're going to do is sit back and do 5 review questions about macromolecules. So pause the video here before we start talking about membranes and do these 5 review questions to see if you understand the structures that we're about to be talking about. All right, so what's the name of the linkage that's formed by dehydration synthesis between a glycerol and a fatty acid? Now, an example of this would be a phospholipid. So this is an example of the bond between the monomers that form together to make the components of a membrane. While glycosidic linkages are found in saccharides, peptide bonds are found in polypeptide chains, which form proteins. And a phosphodiester bond is going to be the component of nucleic acids. So it's going to be an ester linkage that we see forming between this fatty acid tail and this glycerol. Now, the property of phospholipids that allows them to assemble into a waterproof bilayer would be both the hydrophobic tails, which are associating with each other, and these hydrophilic heads. These hydrophilic heads, because they repel or because they enjoy water, are going to be willing to associate with the watery substances both outside and inside of the cell. And the fact that these tails both dislike water actually allows them to like each other. Now, what's the monomer used to build polypeptides? That's an amino acid. The name of the linkage between amino acids is a peptide bond. What's the enzyme that synthesizes these peptide bonds? That would be a ribosome. Which of the following is not a function of sugars in cells? Well, hopefully you recognize the fact that they can be stored in polymers, which store energy. They can be raw materials for other processes, such as being built up into nucleotides or broken down for energy. They can provide structural strength, such as the chains of cellulose. They can be modified on top of proteins, where they can actually be carbohydrates attached to proteins, such as those that are embedded into the membrane, but they can't act as enzymes because enzymes are actually proteins. All right, this is the end of the first component of lecture 4, but I want to emphasize to you guys that even though we're about to start building up components of an animal cell, and we're going to start talking about the functions of organelles, that you should be thinking about the location of each one of these macromolecules, thinking about how they're made, where they're made, what properties they bring to those organelles. So next to this video, I'm going to be posting a bioflix. It's a review that walks you through the structure of the cell. You should watch that bioflix before you proceed on to lecture 4.2, and you should be thinking about what macromolecules there are that are going into those organelles that you see, and make sure that you include those as you take your notes.